is lined by a simple squamous epithelium here, very thin layer of epithelium, which is the simple squamous epithelium, and you have a simple cuboidal epithelium here in your proximal convoluted tubule. You can see it's cuboidal shape here as well. Okay. So what constitutes the brush border of the proximal convoluted tubule and its function? So the brush border has microvilli with glycocalyx, and it is for absorption. Okay, so the next slide will be on the stomach. So it is lined by simple columnar epithelium. You can see here, this is the epithelium. Okay, this whole layer is your epithelium, and these are your columnar epithelium. It's simple because it is not stratified. It doesn't have many layers, just one layer of cell. Sorry, and it's called columnar because it's tall-shaped cells lining the epithelium. Okay, so in terms of description, function, and location, so simple squamous is cells that have the width is greater, meaning they're very flat and horizontally large, or more wide lah. Okay, and mainly the function is for filtration and diffusion. I think I mentioned earlier, because it is thin, it is easy for diffusion to occur across the simple squamous epithelium. Okay, so usually it is for lining of vessel walls and body cavities. So for vessel walls, you want the blood to reach the organs or tissues very quickly. So you want it to pass through a very thin layer of epithelium. Next is simple cuboidal. So they are cuboidal shaped or square shaped. Okay, mainly for protection, secretion, and absorption. So most of your ducts and glands have simple cuboidal epithelium. For columnar, they are taller, okay, taller shaped cells, mainly for protection and secretion. So you have jejunum in the GI tract, fallopian tube, and so on. Okay, you will learn more about the locations during the different systemic blocks lah. Okay, for now, we just want to introduce you to the different types of cell and just the general function and generally where in the body you can find lah. Okay, so jejunum. So jejunum is part of your GI tract. It's in your small intestine, part of your small intestine. Okay, so label the goblet cells. So here, this is the epithelium. Okay, and you have a tall-shaped columnar epithelium. And these are your goblet cells. This rounded shape goblet cells. Can everyone mute their mic? Okay, for now. Okay, thank you. So these are your goblet cells here and here. If we zoom in, this is what the goblet cell looks like. It's more pale staining as compared to the normal other cells. Okay, so what type of gland is the goblet cell? It is a unicellular exocrine gland. It secretes mucus, okay, and it appears empty or pale because mucus is not stained with H&E staining. The staining that we use normally is H&E, and it does not stain mucus, so the goblet cells are less stained, so they appear pale. Okay, so what is the similarity between proximal convoluted tubule of kidney and jejunum? So they both have microvilli with glycocalyx for absorption. Okay, so the next slide is trachea. This is slide of a trachea. So these are columnar epithelium cells here. Okay, you can see they are tall columnar epithelium cells. Okay, but at the same time they have goblet cells as well. You can see here in between the pale staining, goblet cells here in the epithelium. So, and these are your cilia. You can see like a brush, like a toothbrush or brush border lah, but it's made of cilia. Okay, and then basal cells are here, which cells that sit on the basement membrane of the epithelium. Okay, so there are 4 things you need to identify. First, the columnar epithelium here, or columnar cells here, and then you have the basal cells at the base, and you have cilia and goblet cells, which are these big rounded cells, which are more pale staining because of mucus that does not stain well with H&E. Okay. So why is the epithelium called pseudostratified? Because one is that it has the nuclei is located at various levels, so it looks like it has many layers, but it actually all the cells sit on the basement membrane. If it's stratified, not all cell is sitting on the basement membrane, so you have many layers that are not in contact with the basement membrane. However, here all the cells actually sit on the basement membrane, including the basal cells and also the columnar epithelial cells. Okay, so what is the function of the basal cell? It's mainly it is actually made of stem cells to replenish the epithelium. So if the cells here are injured or die off, so these cells, these basal cells will become columnar and replace the injured cells of the columnar epithelium lah. Okay, so where can we find this type of epithelium? Mainly in the respiratory system. So what is the function of cilia? To move fluid and particles along epithelial surfaces. Okay, so next is testis. So this is the complex stratified epithelium. Okay, why is it called complex stratified? Stratified because it's got many layers of cells. Okay, complex because each layer consists of cells at different stages of the meiosis cell division or meiotic cell division or development. Okay, so it's unique for cells in the reproductive system lah. Okay, stratified epithelium, again stratified because it consists of many layers of cells in the epithelium. Complex because each layer consists of different cells of different meiotic cell division. Okay, next is the epididymis. So this is the Stereocilia. Okay, and you can see this is mature sperm which will be absorbed by the cilia. Okay, they are long non-motile processes. The function for Stereocilia is actually for absorption, okay, of residual contents produced but during spermatogenesis. Okay, so cilia is for movement, Stereocilia is non-motile and is for absorption. So next is esophagus. So here, so you need to identify the deepest layer composed of columnar cells, irregular superficial cells which tend to become flattened as they reach the surface, and then a nuclei present in all the cells up to the uppermost layer. Features of stratified squamous non-keratinized epithelium. So you can see here, okay, it is stratified. You have many layers of the is until here. This is your basement membrane here. Okay, so all these are stratified layers of epithelium. So you have more columnar shaped epithelium at the basement, which becomes more flattened as they reach the surface, okay, or the apical part of the epithelium. Okay, and you can see here it is already a squamous type epithelium. Okay, why is it called stratified? Because it consists of more than one layer of cells. squamous because at the superficial part, it is already squamous epithelium or flattened. So we name it according to this layer. Okay, so stratified, squamous, non-keratinized because it doesn't have keratin lah. Okay, usually we find this type of epithelium in wet type of mucosal lining. So for example here, this is an esophagus section, a section of your esophagus, which is a wet type of lining. Okay, so two other sites that we can find non-keratinized epithelium, stratified squamous non-keratinized epithelium, so at the vagina, cornea, and anus. So next, we will look at thick skin. Wait, okay, we will look at thick skin. So it has many layers. Okay, the deepest layer here is composed of columnar cells. Okay, and then we have basophilic layer here, which is about 3 to 5 layers of cells. Okay, which become flattened as they go up. Then we have the stratum corneum, which is this pink line here. And then above it, we have the keratin layer. Okay. So three types of layer in the thick skin. Basophilic means it's more blue or purple. So you can see this layer being more blue or purple. Okay. And it becomes more squamous, approaching the stratum corneum, which is this pink, like very pinkish line as compared to the blue or purplish line below. And the one that is not very compact, which is a bit loose, this is the keratin. Okay, so why do the cells of stratum corneum appear detached? Because they don't have desmosomes in between them. So this is the stratum corneum with the keratin layer. So you can see there are detachments here, like areas where it is not compact. So because they don't have desmosomes in this layer. Okay, so next, the ureter. So our urinary tract is lined by the same epithelium, and it is specific for the urinary tract itself. Okay, because it allows distension of the epithelium when urine is inside the lumen lah. Okay, so when urine passes through or is inside the urinary tract, it can become distensible and it can open up lah. Like this structure is when there is the ureter when there is no urine inside, and it becomes star shaped. But imagine if urine passes through, it can expand and become circular shape when urine needs to pass through. Okay, so this type of epithelium is called transitional epithelium because it can cause this distension and transitional because it can change its shape lah. Okay, so it consists of multi-layered epithelium. You can see here multi-layers. Okay, so this is the apical part and this is the base. Okay, and this is a cross section of a ureter. Okay, and they also have thickened plasmalemma. Plasmalemma is at this at the apical layer here. You can see it's lined by this purple line, and that is the thickened plasmalemma. Okay, the plasmalemma stops the urine from being absorbed inside the cells. Okay, so it's like a barrier lah. Okay, and the transitional epithelium itself, because of its many layers and distensibility, so the function is mainly for protection, barrier, and distensibility lah. Okay, so next is the pancreas. Okay, I'll just bring this down. Okay, so the pancreas is made of exocrine and endocrine parts. So all these acini, these are all acini in the exocrine part of the pancreas, or the main cells of the pancreas. And in the middle here, this is your islets of Langerhans. So islets of Langerhans are cells that produce insulin in your pancreas. Okay, so this is the endocrine part of your pancreas lah. Okay. If you focus on the cells, okay, of the acini, so they are made of basophilia, acidic zymogen granules at the apical end and vesicular nucleus and prominent nucleolus. Okay, this is under low power, so we can't really see the zymogen granules and so on. But if you refer back to my lecture slides, you will see a nicer picture of cells with all these features lah. Basophilia just means that at the base it is more blue or purple. Acidophilic zymogen granules, they have like dots of zymogen granules at the apical end and vesicular nucleus and prominent nucleolus. Prominent nucleolus is a nucleus of the nucleus, and we will see, I think, yeah, here still not prominent because this is under low power, but you can refer back to my slides to see these features, the basophilia, zymogen granules, and prominent nucleolus. Okay, so why do they have basophilia? Because of abundant RER and ribosomes. So these are protein based, and protein stains more blue or purple in the H&E staining. Okay, so because at the acina has a lot of RER and ribosomes, so they stain. They have basophilia, which stains blue or purple. Okay, so if we focus on the pancreatic ducts, we have two types of ducts, intercalated and intralobular. Okay, so the intercalated ducts are here. Okay, so these are intercalated ducts and these are intralobular ducts. Okay, they are both lined by cuboidal, but intercalated ducts are lined by low cuboidal, meaning they are more flat lah. Cuboidal, but they are more flat as compared to the cuboid, the normal cuboidal shape. Okay. So next is your salivary or submandibular gland. They will look the same under the microscope because of the presence of the same type of glands. Okay, so they both have serous and mucous acini. Okay, the serous acini are these purple staining ones. Okay, and the mucous acini are these pale staining acini or glands. Okay, so you can see the difference between serous and mucous acini. So the mucous acini has a larger lumen. In the middle there's a lumen. Okay, here in the serous acini you cannot see the lumen. Okay, and the mucous acini is more pale staining as compared to the serous acini, which has the basophilia because there's more RER and ribosomes. Okay, the mucous acini does not have that feature. Okay, and more flattened nucleus at the side. Okay, you can see here the serous acini has rounded shaped nucleus here at the base, and here the mucous acinus has more flattened shape nucleus at the base. Okay, and these are your striated ducts here. Okay, 1, 2, 3. Okay, so pale staining, mucous acini, more darker staining, serous acini. So what is the modification seen on the striated ducts? It's infoldings of plasma membrane with numerous mitochondria. Actually, we cannot see at this low power lah. If we zoom in only, we can see. So what is the function of the above modification? For active transport of ion and water to concentrate the secretory product, which is saliva. So if you imagine here at the basal surface of the cells lining the striated duct here, you can see there's supposed to be infoldings of plasma membrane here at the base, okay, with numerous mitochondria lah, but very small. You can't see in this low power. Okay, and it allows for active transport of ion and water to concentrate the saliva produced in this by this whole gland lah. Okay, because products of the acinus or the glands will be secreted into the ducts. From the ducts, they will be secreted outside of the gland. Okay, ducts are like tunnels or channels that drain out the products of secretion. Okay, so state the type of secretion of the submandibular gland. So merocrine, exocrine gland, because it secretes the products outside of the gland. Okay, I think that's all for today's discussion. We will learn more in more detail about the histology of these sections or slides or organs in your different systemic modules or blocks. For now, we're just focusing on the epithelium lining of the different, we're giving you different sections to look at the different types of epithelium that we have in the body. Okay, thank you very much. I'll see you on Friday for your exam. Okay.
Thank you, Doctor.
Okay, thank you.
Thank you, Doctor.
Oh, can you take the attendance, please, for practical?
Okay, Doctor. I'll send the link in
Okay,
the group chat. Thank you. Thank you, Doctor.
thank you.
Thank you, Doctor.
Thank you,
Thank you,
Doctor.
Doctor. Welcome. How do I?
Thank you, Doctor.